Welcome back to another new lecture in biotechnology, that is restriction endonuclease. This enzyme is a very important enzyme in biotechnology because they can recognize specific sequences in the DNA and cleave the DNA into fragments, either at specific sites or more randomly. This enzyme was discovered in 1970 by Kelly and Smith and Wilcox in Haemophilus influenzae. This enzyme can be classified into four types like type 1, type 2, type 3 and type 2As. The type 1 is first characterized enzyme and typical example is that from E. coli K12. The active enzyme consists of two restriction subunits, two modification subunits and one recognition subunits. The methylation and cutting reactions both require ATP and S-adenosyl methionine as cofactors. The recognition sequences are quite long with no recognizable features such as symmetry. The enzymes cut unmodified DNA at some distance from the recognition sequence. However, because the methylation reaction is performed by the same enzyme which mediates the cleavage, the target DNA may be modified before it is cut. These features mean that the type 1 systems are of little value of gene manipulation. The most useful restriction modification systems belongs to the type 2. They have number of advantages over type 1 and type 3 systems. First, the restriction and modifications are mediated by separate enzymes. So it is possible to cleave DNA in the absence of modifications. Secondly, the restriction activities do not require cofactors like ATP or S-adenosyl methionine, which should make them very easy to use. Most important of all, the type 2 enzymes recognize a defined symmetrical sequence and cut within it. How the enzymes are named? The naming of these restriction endonuclease enzymes was done by Smith and others in the year 1973. The species name of the host organism is identified by the first letter of the genus name. The first two letters of the specific epithet to generate a three-letter abbreviations. This abbreviation is always written in italics. Where a particular strain has been sourced, then this is identified. When a particular host strain has several different restriction modification systems, these are identified Roman numerals. Most, but not all, the type 2 restriction endonucleases recognize and cleave DNA within particular sequence of either 4 to 8 nucleotides, which have two-fold axis of rotational symmetry. Such sequences are called palindromes. If an object appears identical after a rotation of 180 degrees, that is twice in a 360-degree rotation, it is called two-fold rotation axis. So these are the classification of the restriction endonuclease systems. We have four types: type 1, type 2, type 3 and type 2As. The key features of each enzyme is given in this table. That is, type 1 will have one enzyme with different subunits for recognitions, cleavage and methylation. It recognizes and methylates a single sequence but cleaves the DNA some 1000 base per away. Whereas in type 2, we have two different enzymes. Both recognize the same target sequence that is symmetrical. The two enzymes either cleave or modify the recognition sequence. Type 3, we have one enzyme with two different subunits, one for recognition and modification and one for cleavage. Recognizes and methylates the same sequence but cleaves some 25 base per away. The type 2 is we have two different enzymes, but the recognition sequence is asymmetric. Cleavage occurs on one side of the recognition sequence some 20 base per away. These are the examples of the resistant endonuclease enzymes. You can see it has given three letters name like SMA1. SMA, they are from S for genus name, MA for species name, like Serratia marcescens, like HAE3, which stands for Haemophilus, and A stands for Agapetus, like HIN2, Haemophilus influenzae strain D, likewise BAM-H1, Bacillus amelioidofaciens, strain from H, first enzyme. Likewise, these enzymes cut the DNA into different ways. One, they produce staggered ends, other they produce blunt ends. The staggered ends, the one strand will be protruding, whereas in blunt ends, we don't have any protruding ends. Something like a blunt ends will be getting it, so that is very difficult to ligate. So these things I have discussed already, how it has been named. The first letter from genus, next two letter from species. This is followed by strain, and that is followed by the Roman numerals that indicate the order of discovery. Mostly the restriction endonuclease enzymes recognize the double-stranded DNA sequence that is nearly 4 to 7 base pair long. What is the uses of restriction endonuclease enzyme? They mainly used in DNA mapping, DNA fingerprinting, or DNA technology, DNA sequencing, RFLP analysis, that is restriction fragment length polymorphism analysis, genomic libraries, etc. So we have seen the restriction endonuclease enzyme, where it is present, what is the role, what is the different types of classification, which is mainly used in rDNA technology, how they cut the DNA, and also how they name the restriction endonuclease enzyme, and finally the uses. Hope you understand this topic very well. Thank you. Have a nice day.